your CV. Anyway, Jose will be a witness. Buenos días a todos. Es un gran placer para mí presentarles a la doctora Birte Hocker, quien nos acompaña hoy en este seminario de Frontiers. La doctora Birke estudió biología en la Universidad de Göttingen, en Alemania. Obtuvo su diploma en la misma universidad y después el doctorado en bioquímica en la Universidad de Kohl, en Alemania también. Su primer postdoctorado lo hizo en biología computacional en Duke University en Estados Unidos. Después regresó a Alemania, a Tübingen, como ya investigador en el Departamento de Biología del MPI. En el 2011 se promueve a jefe de grupo también en el MPI en Biología del Desarrollo en la Universidad de Tübingen. Y posteriormente, del 2016 al presente, es profesor de bioquímica en la Universidad de Bayreuth, en Alemania también. Y en su carrera científica ha recibido diferentes honores y reconocimientos. Por ejemplo, es miembro del Comité de Revisión de la Fundación Alemana para Investigación, miembro del Comité Revisor de la Sociedad Alemana de Bioquímica y Biología Molecular. Ha recibido diferentes premios como el ERC, un gran de consolidación para el estudio de las proteínas. Ha sido muy exitosa en conseguir también diferentes grants para mantener su investigación. La invitan muchísimo a organizar diferentes eventos relacionados con la ingeniería de proteínas. Es también miembro de diferentes comités editoriales de revistas muy importantes. Evidentemente, sus publicaciones se encuentran en las revistas más relevantes como Nature, Nature Communication, etcétera, donde ha hecho contribuciones muy relevantes y que, bueno, se van a ver reflejadas en el seminario del día de hoy. Y, bueno, su interés principal es el diseño y evolución de proteínas, entender la relación estructura-función, la ingeniería enzimática, el desarrollo de biosensores, motores moleculares y la biología sintética en general. Entonces, Birte, thank you very much for accepting the invitation and being here today. Please go ahead with your seminar.
All right. Thank you very much, Gustavo. It was a nice words, even though I only half understood them, but thank you for the nice introduction. And yeah, I guess I should probably first start sharing my slides. And hopefully you will be able to see everything now. Does that work? Yeah. Okay. Maybe use a pointer. So that now I can show you my slides. All right. Thank you for coming and welcome. I'd like to tell you a little bit about our recent journey with in protein design. So as I will tell you a little bit about the background and the ideas, what drives our research, I actually called this seminar Protein Design 2.0 because we're living in a very interesting time where there's many new tools coming out and there's actually many developments also based on AI and deep learning methods. And so it really feels like there's new opportunities here. And I had this slide here, this little picture for a long time because it's sort of the dream that we have control of really ordering or fixing certain side chains, for example, in relation to a substrate or something, but that's really tough, and obviously you will never be able to do it like this unless on the computer. So let me get started with what actually drives the questions that we try to answer in our lab. So there are basically what I've always been looking at is at the diversity of proteins that we find in nature. And so I actually like this picture that you have in the background, which is from David Goodsell, who takes natural protein or the information of natural proteins and put it into context of a cell, even though when we work with this as a biochemist, we usually take it out of the cell and look at it in isolation. But basically what I want to illustrate with this is that you have a big diversity of many proteins, whether they're on the surface of the cell, whether they're important for example, for this motor movement of this flagellum that you see here in the space, whether you have actually polypeptide chains that have to fold in order to do all these different functions. There's a huge diversity, and this is all built up out of 20 amino acids, and it's amazing to me. And that means that there are very many different ways you can seem to be able to build proteins, and I'd like to understand that better. So we have always asked ourselves whether there's any information on how these diverse proteins have evolved, and actually whether there are also other possibilities that maybe nature has not sampled, and that we might be able to then try out and learn more about how proteins could be built also in a different way. So with this comes all this question of, can we do this efficiently? Can we really design sequences that will fold into a functional protein and do this efficiently and be able to really build new proteins that either fold in a different way or have new functionalities? And so if this was possible, then we should also be able to design proteins to tackle some of the current challenges that we have. So, I mean, many things, many applications would be in the biotechnological field or in the biomedical field, or if you just think about toxic compounds in the environment, there's so many applications that we can think about. And so if we had robust methods, that would be very helpful. Okay, so this is sort of the motivation that has driving me for the past years in our research, and it really does come down to the fact that we have this paradigm of proteins and how they're built up. And so I'd like to, you probably all know this, but I'd like to start with just reminding ourselves that we have these 20 amino acids, and that the polypeptide chain, which we call the primary structure, is really what determines how the protein will fold, and whether it's secondary structure elements that have to come together to form these very complex globular shapes that then actually can oligomerize and make these very complex, very diverse structures. And so one of the questions that have always been asked and have been a big problem for years is how the sequence actually determines the structure. And so that's what we call the protein folding problem, where we would like to predict how a sequence actually folds into a protein. And this is something that now seems to be at least answered partially, or where there's been huge progress with protein prediction tools like AlphaFold. But then there's also the very intercalated design problem, which actually we call the inverse protein folding problem, where we try to identify a sequence for a predefined structure, or where we say we want to have a certain protein with a certain function, and we need to identify the sequence. So these are two related problems, and that's actually been very fascinating for me to see, and I was really happy to see that last fall now the Nobel Prize was given actually for exactly these two questions. And so on the one hand, there's the prize for David Baker for his work on protein design, really designing novel proteins that had not been seen before in nature, as well as then for Demis Hassabis and John Jumper for the protein structural prediction. And it's just nice to see how this has actually been recognized also in the last year. And so let me kind of give you a view of protein engineering design approaches that the way we see it. So basically, there has been a lot of engineering and design going on for decades. And actually already once we started being able to introduce mutations in DNA and then express in the proteins in cells, that's a way when we could start tinkering with sequences and start engineering. And so this is actually something that has been done a lot, where you start out from a natural protein, where you can introduce mutations and then see how you can change this protein. And you can either do this in a directed way where you actually might have already modeled something. So for example, you could say I take a natural protein and I want to introduce a new binding pocket, and then you might need to optimize it, but optimally you would then end up with a protein that can do a certain reaction. There's also a way to not do this computationally, but actually just take care of this. So basically introducing random mutations and then try to change a protein's function and find a way to make it functional for whatever you're interested in. And so there's other ways as well. And if you that's actually things that we have explored by just using, for example, pieces and trying to recombine them, and then going much higher in the complexity is actually de novo design, really trying to build proteins fully de novo. And this is where I want to tell you most about, but briefly just to put that into a context, that there is a lot of work also going on, and that this is something that is very well feasible. You can basically take natural proteins and repurpose them. And this might actually in some cases be the faster way to do this. And just to name a few, there's, for example, the opportunity to just take a natural protein, and we've done this with a tryptophan repressor, and we changed the binding pocket to bind a very similar ligand. So we only had to introduce a couple of mutations in the binding pocket in order to make this tryptophan repressor recognize auxin, which is a plant hormone. And then we connected these proteins that can now bind to auxin with two fluorescent proteins in a way that once the ligand is bound, there's a conformational change, and that is transmitted so that so the orientation of the two fluorescent proteins towards each other changes, and you can actually have a readout. And you can use these kind of things to build sensors. And so this is something that we have done together with Gerd Jürgens from the Max Planck Institute in Tübingen, where we actually used this approach, where we did this engineering, and they've actually put it in plants where now you can use the sensor to follow where auxin is in a root. And so here you have these in colored, you have the nuclei in which this protein is expressed. And so depending on the presence of auxin, this will light up further. So you will have a fluorescence resonance energy transfer as a readout, and you can follow where auxin is. And if you now move the plant or that root, you can actually see how this shifts. And so these are ways where you can start out from a natural protein, repurpose it, do single point mutation, and you can have really big effects. And for another example where we have been playing around with was actually using an existing protein scaffold or an existing protein like streptavidin and introducing an artificial cofactor in order to make it catalyze a new reaction that has not been catalyzed by streptavidin, by just using this cofactor and then doing several rounds of mutagenesis and X-ray crystallography in order to learn more how to better fix this cofactor in here, and actually by this developing catalysis in such a non-catalytic scaffold. So those are ways that you can do traditional engineering, protein engineering, and really also reach quite some interesting results. If you actually want to make something that is looking quite different, you might still not want to do totally de novo. You might actually want to take pieces of existing proteins, and this is something where we've played around with quite a bit. And that's also something, since I see Jose, hi, he was actually involved, we were working on some of those proteins together. So we have been playing around by taking pieces from, for example, this TIM barrel that's shown up here, and from another protein, which is flavonoxin-like protein, where we saw similarities between these structures, but in the overall structure, you see that they look quite different, and one can do recombination and build chimeras in such a way. And so this is also a way to build new proteins that we are exploring. And if you're interested in these kind of topics, there's a couple of tools that I can recommend to you. So if you're interested in learning more about evolutionary fragments or evolutionary conserved parts in proteins, you can actually use our database of evolutionary conserved fragments that we've built. You can access this via fuzzle on Bayreuth.de and just put in the protein of your interest and see which other ones are similar to this, and browse these similarities. And if you then wanted to actually even use this further, you could even do an automated chimeragenesis with the tool ProtLego, which you can find on GitHub. And there's also an additional analysis tool that's downstream of ProtLego, but you could also use this for any other protein that you're interested in, and that is a protein analysis web server. So just a couple of tools for you that I at least wanted to advertise in case you're interested in using them. I'm always happy to talk about it. But what I really want to focus on today is some of the novel developments, and they are very much actually in the field of de novo protein design. And the idea here is that you can actually try to build proteins from scratch. So basically building proteins in a new way, or actually new ones that have not been seen. And there's the way we got into this was actually And around 2014, when I had been at a conference and I started talking about my love for TIM barrels, which is this fold, you see it in almost all my slides, and the evolution of TIM barrels, and we thought about, well, is there a way to build this de novo? And I started discussing with David Baker, and actually there was someone from UNAM visiting as well, Alejandro Fernandez Velasco, and the three of us actually teamed up to try to build a protein from scratch that is a TIM barrel. And the idea here is that you don't start out from a scaffold, but that you have to think about how this protein needs to look like. And this means that you have to have quite a number of rules how to build this. So for example, a TIM barrel has a central beta barrel. And so you need to think about how the beta strands are ordered next to each other. You know that they are stabilized by hydrogen bonds. So there's a certain geometry that this beta barrel has to have. And based on the length of the beta strands, actually the orientation of your alpha helices is also defined. And so we made at that time, because calculations took a lot, were not feasible on this larger barrels, we actually did it for the 4-fold symmetric one, so that we only had to calculate 1/4 and took that 4 times. So this is what you will see here. We always have a repeat of 2 beta strands with these longer and shorter helix, and the same thing is here then 4 times after each other. And so this is something where we defined what the distances and angles of all of those elements should be, and then built an idealized scaffold and looked for a sequence that could accommodate this. And this was done using Rosetta design. Nowadays we call it the old school Rosetta design, where we then look for the minimum energy of such a sequence. And we were able to, after trying out over 30 different constructs, to actually find one that folded well and where we could solve a crystal structure. And this is just a superposition of those 2 structures. In pink, the X-ray structure, in blue, the model. And we were very happy that we could build something like this, but it's tedious, right? So we had to try a lot of these variants. And then we actually tried to, once we solved the structure, even though it looks very similar, we also saw that there were areas that were not packing exactly well. So we optimized this further in a round of designs. And this is basically the work from Sergio Romero, who's now just returned. He was a postdoc with me, but just returned to UNAM in Mexico City. And he actually really made a really great analysis of all of these different designs, where you can see that they span a large temperature range. So we have designs that actually don't unfold up to 100 degrees. So this is a DSC trace where we do it under pressure, and where we have also a large range of different stabilities, delta Gs, and so on and so forth. And so you can really drive this forward and try to make proteins super stable. The thing that we always thought about, though, is then how to not only make this more stable, but maybe also making this functional and functionalizing this. And so we started playing around trying to extend the loops and whatnot. But then when we were in the middle of this, basically AlphaFold happened and many of the new methods came around. And so I would like to just kind of do a short break here and say, all of these things that I told you about, that is pre-AlphaFold, it's in design. And what we could do up to then was making idealized protein structures, having super short loops. So we often talk about them as rocks, right? We make them super stable, but then that's the structure that they have. So maybe this is not always what you want to have. If you want to have certain functionality, if you might want to have conformational changes and all of this. So that's one of the things. Another thing is that the success rate was really just medium. We had to try a lot of different designs until we got to one that actually worked well. And many of the designs required optimization, multiple rounds of optimization, solving a crystal structure, checking what is wrong, and then trying to optimize further. And as I already said, they are very much just a structure, so no functions were included. At that time, there had been some protein-protein interactions, and very simple enzyme activities could be introduced, but no functions yet. And so this has been changing, and I just want to share with you sort of our experience with using some of those tools. As I was already joking with Jose before, there's too many tools coming out, so we can not really test all of them, but we tried a couple. So where do we stand right now? So let me start by telling you about the first things that we tried, taking our TIM barrel, actually a version that was stabilized with a salt-rich cluster here in the middle. And this is a crystal structure of this one. And we actually thought, can we make this also in a different way? Can we maybe find a sequence that looks very, very different, but can still fold in the same way? And with AlphaFold, you have a quick way to predict structures, so you can use that as a filter. And then there's actually a tool that was released in 2022 here in Science by Ducaras et al. And it's called a message passing neural network that actually is, it tries to find a sequence that matches a protein backbone. And it's based on training data. And so it looks for the most likeliest structure and provides you also a measure of the uncertainty of this predicted structure. And so you basically put in your structure, you let protein MPNN put on a new sequence. You can filter with AlphaFold, see whether it's still predicted to have the same structure. And then you can do this multiple rounds and you can see whether your sequence starts diverging more and more. And so we were looking for sequences that looked very different because we thought how far we can drive this. And but it was a new method. We just wanted to try it out. We had a bachelor student in the lab. That means they are there with us for 3 months. And so Vincent Berg, who did this, he actually expressed 3 designs. And out of the 3, 1 immediately crystallized. So 2 were expressed really well, 1 crystallized. And you can see here now the structure of the starting protein in red with now our design on top. And you can, I think, appreciate that it looks very similar. And so it's quite a different sequence. And so we were just blown away of how well this works. And this is something that I now heard from many other people as well, that protein MPNN really helps you to have a much higher success rate of solubly expressed proteins in E. coli. So there is the training data seems to have really work well for protein MPNN, and by now there's also a whole range of other MPNN-based sequence generators out there. And so compared to our Euler work from 2016, this was really nice to see that design works a lot faster in such a way, or a lot more success. The next tool that I want to briefly mention is actually, and that would be we used, is hallucination. So hallucination, that was published actually by Anishenko in Nature 21. And this uses a neural network, a convolutional neural network. It's called a TR Rosetta. And here you have, so you start out with a random sequence, basically, and you then train it, or you then the sequence is then fed into this TR Rosetta structure prediction network, and then in the beginning your sequence will not really make many connections. So this is now here in the first step of the prediction. Then you have now a contact map where you here have where sequence versus your sequence with very few contacts. And in the beginning, there might not be many contacts, but then you introduce Monte Carlo changes and optimize the sequence always further, funneling it multiple times through TR Rosetta. And at step 40,000, you actually start having really intricate folding, and you come up with a de novo structure. And so this is what you can do with hallucination. You don't really have a way, though, to force it in a certain structure. It just runs wild, so to say, but it's a way to maybe generate new structures. And we thought, this is really highly interesting. Let's give it a try. And so we thought maybe we can use this in order to diversify our TIM barrels. As I said, they're boring. They don't have long loops. So let's see whether we can make long loops. So what we did is we again took our STIM11 with a salt bridge because it's well behaved. And we said, let's open up those loops and let hallucination take its course. Maybe it's going to give us interesting loop structures. And so basically what we did this hallucination pathway, and then we optimized the sequence using protein MPNN, and we filled it again with AlphaFold, and basically found that multiple of those, that we could enlarge multiple of those loops, introduce new sequence. And what was interesting, however, was that it usually always made alpha helices. It always made alpha helical hairpins, which might have been driven by the helix that is already present here, because in most cases it just extends the helix that we have already and builds it longer. We were quite happy about this because we thought that actually gives it more rigidity and more of a stability, and that is a way to actually then generate a nice deep binding pocket. And so we actually picked 6 designs, where three of this first version where we introduced 2 loops, and 3 of the second one where we introduced even 3 of them. So 6 designs were selected, and again, this was working really well. We actually could express them all, and we were able to solve structures of 2 of them. Yeah, okay. If you wonder how we actually filter using AlphaFold, we not only look whether we have a structure, but we actually looked for the PLDTT scores and then a packing of the helices. And we could, yeah, as I said, we could solve the structures of 2 of them, and you can see here now. In green is always the predicted structures, and in color, so here in red or in blue is the crystal structure. And I think you can see nicely that this helix here is nicely extended, the outer helix. Also here in this case it's extended, and in the case of this HaloTIM2, it actually fits fairly well. You have a slightly different orientation of this first helix. And here in this case, the end is not fully resolved, but in general, this worked quite well, matching, making a new pocket right there. In the other case of HaloTIM3, you can see that we're deviating slightly further. So there seemed to be some opening of the structure, but nonetheless, we got, as we expected, these nice extensions. And actually, when you put them right on top of each other, they fit quite well. It's just that once you put them on top of the barrel, they start deviating off each other. So this is really a way how you can generate a little bit of diversity. And this was also working really nicely. But to be honest, I think hallucination is not used that much anymore, because briefly afterwards, there was a new tool that came out, and that is called RF diffusion. And I think that's actually what's being used a lot more these days. And that's something that we also tried out. So we thought, if we can make a binding pocket, the next cool thing would be to actually make a specific binding pocket. Make a binding pocket that is tailored towards a certain reaction. Okay, what kind of reaction would you go for? Well, we thought, let's keep it simple. Let's take a non-natural reaction that everybody else also uses. It's a Kemp elimination. And basically said, what about we take our de novo TIM barrel, and now we say we want to have catalytic residues in relation to a substrate, and now we build a custom lid on top of it. Because if we place one catalytic residue in our ligand, then there's not really space to fix the second residue, right? So we might want to have structure that is able to provide the second catalytic residue from the top. And so this is something that we now did. Let me walk you through the way we tried to design this. So this we actually started about a little bit over a year ago, when Roberto Chica from University of Ottawa was visiting us. And so we teamed up and said, let's try this together. And here's again the reaction, easier to see for you. So here you have your this is a Kemp reaction where you have your benzoxazole, and there is actually base abstraction from this side, and then from the other side, it's nice to actually to stabilize the site and have a charge to change that charge distribution to the second residue, but it might not actually be fully always necessary, but it can actually improve that reaction. So there's 2 residues that we have to place. You can actually choose also others than aspartate, but that's the ones that we chose. And the other nice thing is that you can easily follow that reaction, because upon the ring opening, you have an absorption at 380, and this compound is nicely observable. The other thing is, it's a single-step reaction. It has a fairly low activation barrier, and since it's a non-natural occurring reaction, you don't have any trouble with contaminations usually. Okay, so we said, let's see whether it can make maybe a lid that allows us to make our fairly boring de novo TIM barrels into enzyme-like TIM barrels. So what do we need to do for this? We used a combination of physics and the deep learning-based approaches. So the first thing is that we needed to place our transition state, which defines the combination of the side chains, the catalytic residues, towards the ligand. And here you see now, basically trying to put it into the context of a barrel, we can find a placement for the first catalytic residue, and this defines where the transition state has to be, and then the second residue has to be in the right orientation. And once we do it, it actually ends up being somewhere in the air, right? So there's no structure present. And so that's what we then took as a point from which has to be there in the structure generation. And so for this, we use this Rosetta fold diffusion, which is called RF diffusion. And so in this RF diffusion, basically it takes initial Gaussian noise and then recovers it into a three-dimensional structure. And if you so a way to illustrate this is here, where you have this noise that then it will be combined into a structure where you, for example, have here like a beta hairpin and additional helical structures. So we introduced this also by opening up multiple of the loops and introducing again random noise here and generating a structure. This structure, however, doesn't have any side chains. So it only has glycines. And we know that glycines will likely not form the structure. So we have to find now the right sequence to generate this kind of structure. And so for this, you can use, as I showed you before, protein MPNN, right, for sequence generation. So you start out from this glycines here in this stretch in the loops, and then you use protein MPNN to get suggestions for sequences. So that's the third step. And then you actually want to check whether this is still likely to form a structure. So before going to the lab, we actually just say, let's take AlphaFold and see what it would suggest us. And then we can compare our the diffusion model with the AlphaFold predicted model. And if it really nicely overlays, then that's a good hit. That's a good one that we want to start out from. So let me tell you, we started out with actually lots of structures. There were also some funky ones in there that we then filtered out. And especially then by this AlphaFold step, then you start picking out a couple of structures with whom to continue. And the next step, so these were all the deep learning methods that are new out there. In this last step, we then said, okay, let's use physics-based again, because there had not been so much about packing a small molecule. And so there's more experience with physics-based approaches. And so for this, we used the program Triad that had been that is a, well, you could also use Rosetta actually, but it's a physics-based model that uses this also that elimination and pairwise decomposition of interactions. And so we used this to place again the ligand, because after AlphaFold, you don't have this ligand, the transition state in there anymore. So we placed it again. We looked at side chains in the vicinity in order to pack it better. And for packing, we then used for the repacking of the active site and to generate a library of different side chains, we actually used now Triad. Okay, and then of those, we scored all of those based on the Triad scores, and we picked a few chemtims to test experimentally. And so Roberto did this for a few of his designs, and we did this for this. And actually, so there's a number of people involved. So on our side was Julian Beck and Emily Freund. And we did this in her master's thesis. And then here's Roberto with PhD student Yayesh and also an undergraduate student Ben. And so basically, we were happy that with the 4 designs that we tested, so we actually only did 4. We had one good hit, and that is shown here. So this is your substrate, and you can see that, I'm sorry, the resolution is not as I expected, but I hope you can see well that that you actually get a nice kinetic from which we can not only calculate our kcat, but also our kcat over km, actually this is not a great enzyme, but it is doing obviously turnover, and it's doing it in a similar manner as other de novo designed chem eliminases that have been done before are, but all of those other ones, they don't have new structures. So we were now able to put on a new structure, and it seems to be really dependent on this, because if we knock out the 2 catalytic residues of ChemTim4, this activity is gone. And we did another round on top of ChemTim4, where we actually used now ligand MPNN, and which led us to ChemTim4-2. And that one is now, it's behaving slightly different, which is interesting, where we can only get the Kcat over Km, which is in a similar range, but it's behaving slightly different. And at the moment, we are analyzing its structure, and it would be really nice to see how exactly this is folding. And so it really looks as that with deep learning, this enables the design of new enzyme reactions. And there's lots of papers coming out. I think there's just a few days ago, there was also one on serine hydrolases. And so I think with these tools, at least in our hands, it also seems to be working fine to really try to make new structures that can accommodate active sites. So that means that if there's all of these learning models, I would like to ask the question, is there maybe something that's inherent in the sequence? And sort of asking the question, is there a language of proteins? And so this leads me to another path that we have been trying to learn more about sequences, because we've always been doing all these sequence alignments and looking into the evolution. So there's actually something that we tried also using deep learning that's a very different approach to the ones that I've been showing you up to now, because the ones so far always have been dealing with the structure first. Now this is more of a sequence first approach. And that has to do with the fact that we saw similarities, we kind of see similarities between sequences and language. And so already going back to the Anfinsen's principle, where you say that in the sequence, all the information should be in order for the protein to fold, we think, or we hope that all of that information can be somehow learned. And so if you compare actually the different levels of structure in proteins to language, you see interesting analogies. So the amino acids are analogous to the alphabet. I guess that's simple to see. So if you then put a couple of amino acids together, you can actually form beta strands, alpha helices, certain loops. So in a similar way, when you put letters together, you make words. And so one could say words are analogous to these secondary structure elements. Once you put those secondary structure elements together and you form your tertiary structure, this is where you start having interactions, where you start forming binding pockets or interaction surfaces. And this is usually important for function. And so that's what we see as analogous to when you put words together, that makes a sentence, because then you have a certain meaning and you have a relationship of some words to another, similar to how you have the relation of folding of a certain secondary structure element towards another one. And then the next level would be the quaternary structure, where you build up complexity, which is similar to building more text out of multiple sentences. And so you can increase this this complexity quite nicely. And that's something that Noelia Firuz, who was a postdoc in my lab, realized quite nicely. And she said, okay, why not use this language models actually also for proteins, because she was seeing this similarities. And so she basically took a data set of sequences from UniRef, downloaded this, and then she took, there's about 49 million sequences. She took 10% for evaluation later on. She didn't use them, but the rest she used for training. And she trained a transformer model in order to, using actually the same set that you might know from ChatGPT. So this is a pre version of it. It was GPT-2. And she trained basically on this model on the data set of these proteins sequences at the Erlangen cluster, where we got access for a couple of days, and where she could actually train a fairly large language model, which then generally made this transformer that you can now use to generate sequences. So oh yeah, I don't have a slide for this, but basically what you can now do with this transformer is you give it a prompt, like a methionine or a short stretch of amino acid sequence, and then it keeps filling in. So what is the most likely sequence that could come after? And so you can generate sequences of different lengths. And so this is something that we then did using this transformer, which is called ProtGPT-2. And you can generate many, many sequences. And here is a data set of 10K sequences that we generated. And then we looked into this data set, how many of these would be predicted to have globular domains, for example, or ordered and disordered content. You can also do predictions for secondary structure, like alpha helical content, beta sheet content, foil content. And this is in very similar ranges as is when you take natural proteins. They have similar amounts of globular domain, similar amount of ordered content, and so on and so forth. So that was very interesting to see. And so basically, what you can then do is you can see whether these sequences are actually similar to natural sequences. And this is what you can now see in this central part of the slide, where basically we made a cluster map of all of those natural proteins that are known from the SCOP database. So this is SCOP is this structural classification of proteins, where you have all alpha proteins, for example, in orange. You have all beta in light blue, dark blue for alpha/beta, and so on and so forth. And actually, the PROJECT-2 generated sequences are all the ones in white. And what you can see here is that they are all over the place. So they sometimes cluster with helical proteins, sometimes with beta sheets, sometimes with mixed ones. And so that means that PROJECT-2 is not just generating one kind of sequence, but many different kinds that nonetheless seem to be in some ways similar to natural proteins. So in the center, we have many that have similarities. At the outside, you have sequences that are not so similar. And now if you look at some of those in more detail, and you do structure prediction with AlphaFold, for example, then you get these kind of structures that I show here. And for me, it was striking to see that these structures look very nature-like. So they do have good packing. They have all types of folds. So we have my favorite Tim barrel, but we also have all beta structures or all alpha structures and all of these mixed types. And actually, they have also very good PLDDT scores. So for example, up here, we have a PLDDT from 94, which is really high. And so this is looking really good. And despite this good this good prediction, it's actually not necessarily that similar to natural proteins. So for this protein, the closest one we find is with this PDB identifier, where you have 26% sequence identity. But there's even some where you have even lower sequence identity. And somewhere I would say, well, that's hardly anything, right? 9% is the same as random. And so there seem to be maybe really new ways or enlarging the sequence space with which you can access these kind of structures. So this is what we've been fascinated with, and we've been trying some out. And so expressing over 100 of these sequences, and many of them express in E. coli, even though not all natural proteins would necessarily express. So I think 68% is actually quite fine. Many of them are not soluble. They end up in the insoluble fraction. So about 20% we can easily purify from the crude extract of E. coli, and we do get quite good yields. What we're still working on is trying to solve structures of these. That hasn't worked yet so far, but we are hopeful that this will happen soon. Okay, oh yeah, this is just more or less of the same. So let me now talk about talk about another way of making proteins with language models such as PROJECT-2, or actually another language model that Noelia now developed, which is called Sim Control. She published the theory in this NeurIPS report. And so actually by now there's quite a number of language models, and they can differ in terms of being conditional or unconditional sequence generation. So for PROJECT-2, that's unconditional. It just generates sequences, right? But what we would like to do in some cases is generate sequences that have a certain function. So you probably would like to get sequences that can do something interesting. And we thought, let's try these models to make sequences that are enzymes. And let's take an enzyme that is really interesting for us, and that is a central enzyme from glycolysis, which incidentally is also a Tim barrel, namely the name giver, the triphosphate isomerase, which interconverts GAP into DHAP. And so we started out with Sim Control, where basically this model is trained on the enzyme classifications, which is shown here, so the EC classes. And so you can basically tell it, give me sequences that have this EC class, which actually fits to triphosphate isomerases. So that's our prompt. And then we generate sequences with, which we call spoken TIM. So let's look at these first. Yeah, this is just to show again why we use triphosphate isomerase as a model system. It's actually a very complex model system because it's a dimer, and the active site is completed by actually by a loop that swaps over from the second chain, and so it's supposed to be an obligate dimer for function. Although there are also monomeric versions that work. And it's yes, I said it's an important enzyme in energy production. It's really ubiquitous. You find it in all domains of life, and it's highly efficient because it's called to be diffusion limited, because every time the substrate meets the enzyme, there is conversion. And the good thing is that there's lots and lots of sequences available, and that's good for the training. So we thought, let's take this as an example. And so this is something that Sergio did when he was still with me, and working with Alex, who was a master's student at that time. And so they generated lots of sequences, 90,000 sequences, and filtered them. And the first thing that what you can do is you can actually compare the sequences that you get with Zim control here in blue to natural TIMs. And so the interesting thing is that there are some TIMs that are shorter than we expected. So we're actually not entirely sure whether all the sequences that are in the database, whether they are actually fully functional, because some of them seem to be shortened. So we didn't really want to have these short versions, which you also have with the natural TIMs, and we focused on sequences that are more in the range that we expect. So we said we did a first shallow filtering by saying we only want sequences that are in the length of 200 to 300 amino acids. So we basically take everything that's above this. The distribution is very similar of our Zim control generated sequences to the ones of the natural proteins. So that we thought was very interesting, and so we thought this seems to be a good set. We also then selected only sequences that were low in identity. So we said we don't want to have anything that's above 35%. So we wanted to get further away. So we are actually entering sort of that twilight zone of sequences and said, let's just take those sequences here. And then the other thing that we did was, for those sequences, we predicted with AlphaFold multimer the structures, and we looked whether we could get a good dimer in which actually the active site is present, where all the catalytic residues are present, these three, as well as a loop that is reaching over. And so for this, that means for these, we actually took 12 sequences that we chose for experimental characterization. And these we call spoken TIMs. And the first thing that we did is that we actually checked whether they're able to complement triphosphate isomerase deficient strain. So just see whether they're able to, if you knock out the wild-type triphosphate isomerase, whether you can actually complement. And so here are the controls in this first in this first plate, where you have, so we let them grow here on glycerol, where you have if you have only your plasmid, then nothing should grow, and that's the case. And if we take triphosphate isomerase from two different organisms, we actually get complementation. And in the case of CP-TIM, you can see it even more nicely than for the SP-TIM, but they all both grow overnight. And then when we look at our spoken TIMs, you can actually see that number 5, there seem to be a few very slowly growing colonies, but the best one is actually really spoken TIM 9. So out of the 12 that we tested, we have one that really is able to complement very fast. So we got really excited about this and decided to look at this in more detail. So we purified spoken TIM 9. The yields are okay-ish, could be better, but we can purify it. And then we can do actually mechanism kinetics, and we can actually deduce the catalytic efficiency. And that's basically here. You can see this nicely, that we have a really nice turnover, and it's very fast in comparison to our Kemp TIMs. This is way, way faster. So basically we have pKa of 7.3 times 10 to the 6 per molar in second. And if we compare this to E. coli TIM, this is actually only 30-fold less. Well, so it's really in a similar range, I would say. We were super excited to see that apparently the SIM control can generate a highly active TIM-like sequence. And so if we knock out the catalytic residues, so that's our other control, we knock out the catalytic residues, then we actually don't have any activity. That is here the blue line, and also we don't have any growth in with a TIM deficient strain on glycerol. So in vivo and in vitro, we can show kind of this activity, and we can actually see that it's dependent on those residues that we introduced. We also did a little bit of biophysical characterization. So this is just a circular dichroism spectrum where you see that we have the typical CD spectrum for a mixed alpha beta protein. We actually also looked into, so we did size exclusion chromatography connected to multi-angle light scattering. And so we do see that there are two different conformations. So one of them is, this would be mostly corresponding to monomer, but there's also dimer, so there seems to be an equilibrium between these. So maybe that we don't. I mean, that's really this should only be the active compound, and so maybe we are losing some of our protein and actually have even higher activity for the dimer phase, but the dimer might not be as stably formed. And then when we do unfolding studies using DSC, differential scanning calorimetry, we actually do see that there's two transitions which could be actually also have to do with a monomer dimer. And so this is a really interesting case, and we would like to solve the structure of this protein, but we actually this is really difficult because of getting it into high concentrations. But what's super exciting is that it can really it really shows all the evidence of triphosphate isomerase activity. And so one thing that you can then look at is whether the interactions, even though we don't have a crystal structure, what the interactions would look like in an AlphaFold model. And so actually it really looks like that the language model is able to generate interactions all over the place. It's not only the residues that are close to each other, but they actually can make interactions of residues that are far away. So for example, we have the salt bridge cluster of residues that are quite far away in sequence and that are predicted to be or that are suggested in this design, and they should really stabilize the protein. There are also hydrogen bonding networks that we can see here nicely. We have hydrophobic clusters that are formed, high stacking interactions, all of this. And so really this the sequence that gave us this active enzyme looks very native-like. So now I should explain to you the other arm of this generation. So I talked about the SIM control. We also tried to use the unconditional model, ProtGPT2. And in this case, because it's unconditional, you have to actually fine-tune it. And we fine-tuned it with 1,000 natural triphosphate isomerases from the sequences that we had, and fine-tuned this model and generated then sequences with ProtGPT2. So these ones we call GPT-TIMs, and there's filterings where we actually looked for net charge of sequence, spatial aggregation propensity scores, and also in the predictions of the solvent accessible surface area, and so on and so forth. And so for this, we also chose 12 designs, and this is now the complementation that we did here. And so here you can see the controls again, only the empty plasmid, but then here are our controls. So this now, in this case, I'm showing this in glucose. So here's another control that grew nicely, and here you have now one variant, GPT2, which really complements also very fast, as well as GPT-TIM2. So there are actually even two candidates that seem to be able to complement the TIM deficiency and be able to do and be nice enzymes. Unfortunately, so far, these are not easily expressible, so they seem to be very efficient, but we're still working on getting enough of the protein to do also an in-depth in vitro analysis. And so when we look at the way of filtering these methods, that's where we actually have differences. So for spoken TIMs, we actually start with, in the initial case, with the initial sequences, we can actually look at the residues and look at how often we find certain residues in this area. And here we have actually catalytic residues that are very important, but also the vicinity seems to be already very similar. And then we only do a very shallow filtering where we use the sequence length, so this doesn't really change. And then the final selected sequences, there's a little bit of a difference because we used the AlphaFold multimer, but basically we already had most of this logo present in the beginning. The GPT TIMs, on the other hand, the ones that are generated, they're very diverse and very different. There's actually quite a number have proteins in here. And so this is what we then had to, so here we also did sequence filtering, sequence length filtering that already changed that logo quite a bit. And then a very important one was this advanced filtering, which I had said before, that we actually were looking also at all these different scores, and that was needed because otherwise we actually we probably would have had more that didn't really work as well. And so the filtering increased our maybe our success rates. And that's just something that we can do by looking at the sequence you get out at each of the steps. And so this can help us to actually optimize the output. And that's something that we were able to do because we first did the spoken TIMs, and then we could actually check whether we are now in we now get similar output once we do the filtering. And that's been working really nicely. And so with that, I should come to an end. And that is that really I feel that there's lots of new tools out there, and that this is really exciting time to not only build structures, but actually build also proteins with certain functions. And so I really feel that there's a new era of protein design happening. And so there's on the one hand really the high success rate that you get with protein MPNN, but also you can build new non-idealized structures with either hallucination or RF diffusion and make them even custom for design of enzymes. But there's also a parallel path that's really interesting, and that is designing from sequence directly to design proteins or enzymes using these large language models. And you can use either conditional and fine-tuned unconditional models to generate new enzymes. And so really, this opens up so many new possibilities for protein design. And I hope that with showing you our experience or our samples, that this might be helpful for you to also apply these methods. So this is a fairly recent picture of our group, and some people that I want to highlight is actually in particular Julian, who tried out many of those methods. I mean, he did the hallucination methods, also the RF diffusion work together with Emily for the Kemp TIMs. And then we have here Alex, and then also Sergio, who really worked a lot on the TIM reactions to the large language models, and Noelia, who trained the original transformer and got us into all of this. And the current team, all the alumni who I'm always very happy to connect with, and our collaborators as well as funding sources. And thank you for listening. Open for any questions.
Yeah, thank you very much for your presentation. So for the questions here in Zoom, please just raise your hand and we'll open the microphone. And for the people in YouTube, please write down your questions and we'll read them here. So questions?
I only see hands. I don't know. Gustavo, you do this?
Yes, I do, but please write the hand in the application so I can see better.
Jose, do I need to stop sharing?
If you want, yeah, that's okay. Or maybe you will have to go back to some of the slides. So
because I don't see the chat.
Go ahead. Okay.
I don't know where it went, the chat.
I have a question, but I mean, I don't know if I want. I mean, everybody wants to ask probably.
Yeah, but so far only you and Lorenzo raised their hands. So go ahead.
Perfect. Okay, perfect. So, I mean, I have several questions, but I'm like more in general, like there are two questions, like maybe I can be satisfied only with this. So how do you, I mean, in general, how do you do the scoring of the models? Like, do you always include the Rosetta's energy function to score? Or is mainly just using the machine learning models? And the second question is, have you managed to get betas with RF diffusion?
Yeah, two good questions. So, I mean, the Rosetta energy units, that's what you get with the, that's with the old Rosetta. Oh, now I stopped sharing, I guess. I did it automatically. Okay. Basically, yeah, you get, it depends on your tool that you have, but basically for the design for the chemtims, we in the end, so we use different steps, right? So there you use, on the one hand, you get the sequence protein MPNN, but then we actually score later with TRIAD, in fact, and we looked into the structural similarities. So there's multiple steps where we filter. For the large language models, that should be inherent. There you actually don't have such a score. So it really depends on the tool. So that's the first question. The second one, beta strands. Yes, because it was really interesting to see that the hallucination gave us all these alpha helices, and it also seems that there's many more alpha helical structures that are found, and maybe that has to do with the fact that once you have a helix, it's easy to just continue that, right? But in the case of the chemtim, we actually had also a beta hairpin, and we looked for this and took it further because we thought it was interesting, and in some cases we do also find beta sheets, and so that's yeah, but it seems to be helices are a bit more present.
Prominent. Yeah, thanks.
All right, so Lorenzo, you had a question.
Yes, hello, very nice talk again. I have a question about the flexibility of all your designs, because one of the important things to have an enzyme is to have dynamics in it so that it can have quick turnovers and stuff like that. Can you think of a way of codifying that in your large language models or stuff like that, or you just need to screen for them to find just by serendipity something that is faster?
Yeah, so honestly, I think that probably we already have this inherent in the models. So I think, and if I just look at the designs that we have from the large language model, we actually seem to have proteins that are a little more floppy, that are a little more able to go between different states. You know, so even though we have this high activity of spoken TIM9, it is in an equilibrium of monomer and dimer, and there's probably is some of this conformational already in there. So basically, since we take all the information from the sequence, the only hope I can have is that it actually is coming from the sequence, because there's no other way to introduce this. But we have been talking about this a lot in the context of the chemtims that we have designed using RF diffusion, protein MPNN, and then TRIAD. And there, we actually have looked into a design from Roberto that was not really, that didn't show any activity, but that actually seemed in MD simulation, we actually saw that it seems to be forming this structure. And in some ways, we are even wondering whether we sometimes maybe make things too stable. And because you need to take care of access to a pocket and in order to have your product, your substrate come in, your product be released. And these are things that certainly we need to take into considerations. And so there are different ways that one can think about it. There was a paper from Gustav Oberdorfer where they actually reported also that they've made tunnels now extra. They paid attention to actually keep access. And that's one idea to do this. It's not conformational changes, as you say, but it's at least taking that into consideration. And so, yeah, dynamics is really an important next step. So what some people are doing, or what one could actually do, is one could select for different conformations and try to do that. So yeah, I think that's one way. I hope that answers.
Yeah, perfect. Thank you.
And now start seeing the
There are questions in the chat. Do you want to read them?
Yeah. So there's one. Alejandra is asking, I wonder whether the new TPIs enzymes are also subjected to post-translation modifications as a wild type. Yeah, you know, we just expressed in E. coli. I don't know how much this actually is happening. We haven't really checked it. It's a good question. That's maybe something we need to look into. At the moment, I don't really expect this, but sure, it could be the case. So then there's another question from YouTube from Nuria Sanchez. What would be your advice on how to choose the starting scaffold of a protein to do design and evolution on it? Oh, well, that's a good point. I mean, it gets, I think it depends very much on what you want to do. So I always had this, and that's why I mentioned it at the beginning, that you can often just start out with a protein already. I always look for something that does something similar already. For example, if I want to bind a small molecule, because binding small molecules to design that is not very simple, so I look for something that looks similar, so that I can take advantage of part of the binding pocket. That's also the idea when I take fragments, you know, that you can already have a partial binding pocket. So this would be one thing I would look into. The other thing is you need to be able to use this protein in the lab. So a very important part is thinking about experimental details. Is this really well expressed? Is this really something where I have a good assay to do this? So there's a lot of these practical issues that I would think about. And then for the evolution, you need to have you need to think about how you're gonna select, or whether you can select, or whether you have to screen. So for example, for the triphosphate isomerase, that's really nice that we have the deletion strain, so we can just we should be able to now do just direct evolution, right? So this is always very handy. So maybe those are things to think about. There's another question from YouTube from Miguel Angel. Do you know an example of an enhanced enzyme using an AI? Usually AI designs are not as good as natural enzymes. Even more, direct evolution appears to be more useful. Well, honestly, I don't think just with directed evolution. I mean, I'm already pretty impressed with the TIM that we got, the spoken TIM 9. It really seems to be quite active. So it's not as active as a natural one, that's true, but it's really not far from it. So in this case, I think we could make a really big leap. With a combination of Rosetta fold design and then protein MPNN, yes, our ChemTim is not maybe that great yet, but there's I think that has to do maybe with optimizing those methods. On the other hand, with direct evolution, you don't get the starting point. So it has always been the argument that you can use computational design to at least get a way in. Even if you make a crappy enzyme, at least you have something on which you can evolve. If you don't have a starting point, there's nothing to evolve on. So I think that has already been useful, but maybe now there's even better ways to do it. And maybe we can actually optimize and train some of those methodologies to give us even better enzymes. So I don't know, I haven't given up hope on that. And I think direct evolution is immensely useful, but it does something different. And so I think what's the most powerful is combining all of those methods. And we have all of them at hand, so why not use them together? They shouldn't be competition. They should really be, we should really take advantage of it, that we can combine them. So I hope I can answer the ones from YouTube because they're not here. Happy to answer any other questions you might have.
Yeah, any other questions?
Well, I have another one. So like, especially the case when you're designing this to the dimer with the catalytic loop, how do you score the interaction surfaces between the two proteins as well or not?
So we looked into, so we really had this very shallow filtering with spoken TIMs. So there we only looked for the sequence and then for the structure prediction. So it's a structure prediction whether you have that loop being in a similar area. So you can do just an RMSD.
Right.
So it's not nothing more there. And and then we had this additional filtering in the GPT TIMs. There we looked at the scores that I just put there. So it's more surface accessible, surface area, all the catalytic residues there. These are the things, but it's not really that much that we have to filter out. We already seem to be getting quite good sequences from the start.
All right.
Yeah, it changes a lot now with so if you would design the structures, then you would, we always used to look at all of those details, but with the language models, it just seems to be taking care of quite a lot of these things already.
And maybe the last one, also like in the case of when you were diffusing just the specific areas of this protein. So did you have to change a lot in the code from RF diffusion?
No, you
or how do you?
No. That's something that you didn't have to really. Did you have trouble with that or?
No, I just was wondering how did you do it? Like just to diffuse specific areas, no? Not the whole protein.
Yeah, basically, you can define which areas you want to diffuse as. So yeah, so it's true that many of the, if you look at the code or at the papers where RF diffusion is shown, it's often that they just make an entire new scaffold. That's another option. So I guess we were a bit careful, and we thought we already have the de novo TIM barrel that's built on top of that. But there's definitely also a push towards saying, why not do everything de novo?
Right. And then just the last question in the technical details of how do you run the things? Did you buy GPUs or how did you do
you
it?
have, I just look at your desktop computer. You probably already have a quite good CPU there. So there's quite a number of things you can run already on your desktop, but we also have access to a couple of GPUs. You probably also have, I could imagine you probably also have some clusters at CLC's.
I don't know.
Yeah. So, but the
Okay.
GPUs, they make it faster. Yes.
Great. Thanks.
So any other question?
Julia?
I would have one question. Thanks for the nice talk. In response to Jose, or in a question, how long are taking, for example, these calculations? When you say we can run it on a laptop, how long are basically these calculations taking? Are we talking about days or weeks?
No, days. So it's also the question,
okay.
yeah. So it's also a question of how many you want to generate. So in the end, it's also, I mean, just generating a few, that doesn't take long. It's a question whether you want to really generate many of them. So that's what then takes longer. But otherwise, no. What took really long was training that model, or rather, that took a lot of resources. So actually training that first transformer, that's really where we took advantage of having access to this cluster at Erlangen University, where we had 4 days on multiple GPUs. But then once you have the transformer, then it doesn't really need that much anymore. So the training is what takes a lot.
Okay, the training is the time-consuming part in this case. Okay.
Yes, and resources.
Thank you very much.
Yeah, welcome.
Okay. Thank you very much for your very nice presentation, and yesterday with the students and today with the whole community in CCG and IBT. Thank you really for your time.
Yeah, thank you for listening, and it's been good seeing people again. Nice to see you, Lorenzo.
See you. Bye-bye.
And yeah,
Bye.
thank you. Take care.
Bye-bye.
Bye.